Hi. In the upcoming segment, we're going to continue with quantitative biology and sometimes referred to as biology by numbers to discuss a little bit the census of biomolecules at a cellular scale. The numbers that I'm going to give you, I will discuss a bit about whether we need to remember them, why, what sense it makes, and which ones are more important than others. But as I stated at the beginning of the segment, any number has to be against or for some hypothesis or model. One of the important aspects of getting numbers is how we measure them. So experimental techniques, become very important. We will also speak after this about time scales in terms of movement, growth, and transport, and discuss a little bit about some very fascinating questions about how cells manage to keep regularity in time. They're almost like clocks. They work, in fact, in many senses, like clocks. But they're not clocks, obviously. They don't have the wheels, they don't have the digital machinery. So how does that function? We'll talk about it. And finally, we will end this order of magnitude and biology by numbers segment by discussing energy scales. Because no matter how we see it, energy rules. And this is sort of going to be our next segment anyway. So let's get to it. You're looking at a table taken from a textbook, Physical Biology of the Cell. And perhaps those of you who studied chemistry or physics are aware that typically physical or chemical constants are mentioned at the beginning of the textbook. I challenge you to show me a textbook in biology that does this. And if you are a biologist, you should argue that this is not easy, because if I show you a microbiology textbook versus a biochemistry textbook versus an animal physiology textbook, the numbers, the variables that are used, as well as the values that are found will differ. And it is for exactly this reason that we spent some time thinking about E. coli as a model cell. Because what it tells us is that at least for one cell type, can we get some numbers that allow us to get a relative sense of magnitude, right? So for example, if I look at the first of these variables, which is with regard to E. coli, and we take the numbers for volume, then if you recall, we described E. coli as a cylinder, spherocylinder with some ends of 2 micron by 1 micron. But if we decide to ignore all this detail, then we can perfectly well assume that it's a cube with a length of approximately 1 micron. This is what we are referring to when we talk about scale. In other words, relatively, how big is it? And therefore 1 micron cube. The mass of an E. coli cell comes out to be 1 picogram approximately. We say approximately because, amongst other things, it may depend what cell cycle stage we take, what growth conditions we use, what bacteria bacterial metabolic state we are looking at, and so on. So also to remind ourselves, what is 1 picogram? 10 to the power minus 3 grams is 1 milligram, minus 6 grams is 1 microgram, minus 9 grams is 1 nanogram, 10 to the power minus 12 grams is 1 picogram. So this is nothing but 10 to the power minus 12 grams. Cell cycle time is about 33,000 seconds. Those of you who studied microbiology maybe know that we typically state that the so-called doubling time of E. coli at fast growth is 20 minutes. This 3000 is nothing but 50 minutes. 3000 seconds is 50 minutes. We are again trying to consider average times. It becomes an interesting problem, which is to ask, in terms of the cell cycle time of E. coli, how long does it actually take to divide? There is a law which goes by the name of the person who came up with it, Schechter, Moselio Schechter. He writes a very beautiful blog on American Society of Microbiology's website, which states that the growth rate and cell length scale linearly. This is called Schechter's law, and there's a precise relation for this. So in a way, these are not independent variables. They are interdependent, you could say. So by changing one, you can change the other. And the causal links, you see, once we put a number to it, the causal links become interesting. What about cell surface area? We can come up with 6 microns square, which is considering the spherocylinder for the cylindrical part. We have pi r square h, which if I take my calculator is pi into 0.5 r square, pi 0.5 square, micron square, pi into 0.5 micron square into 2 microns, which is 1.57, about 1.6 micron square. And the cylinder has spheres on top, so we can add that volume, and we are referring to the volume of the surface area. This is 1.5 micron square. We're referring to the area. The area of the caps is 2 caps. We can argue they are hemispheres. So 2 caps make 1 complete sphere with radius 0.5. So we're talking 4/3 pi r, 2 pi r square. 2 pi into 0.5 square. It's 1.5 plus 1.5. It's 6 micron square. I want you to find out how the cell surface of an E. coli cell can become 6 micron square. It's important at this point to question these numbers because they are approximations, as I keep highlighting over here, but these approximations are obtained from certain assumptions. What about macromolecular concentration in cytoplasm? It's about 300 mg per ml. Genome length is 4.8 into 10 to the power 6 base pairs, which is approximately 5 into 10 to the power 6, 5 million base pairs. Swimming speeds is about 20 micrometers per second. We discussed this in the earlier section on fluids. So should you remember these numbers? Absolutely. Why should you remember these numbers? Because as I was discussing, these are telling us about the scale and relative size of things at a cellular level, which in fact for almost no other organism do we have with such detail and such reproducibility. Which is another way of saying that we would love to have these measurements for other cell types too, but until we have them, we're better off with the numbers we have. So yes, you do need to. What about bigger cells? Yeast is a eukaryote. This is Saccharomyces cerevisiae, the brewer's yeast or baker's yeast. It has a volume of 60 into 10 to the power 3 micrometer cube. The mass of the cell is 60 picograms. Remember, it's about 60 times that of the E. coli cell. The diameter of the cell is 5 microns as opposed to 1 and 2 micron for E. coli. And cell cycle time is 200 minutes as opposed to 50 minutes for E. coli. Everything is bigger, everything is slower, and the genome is 10 times longer, which is 10 to the power 7 base pairs. Organelles, on the other hand, like the nucleus, cell nucleus, are of size typically 5 micron diameter, but they may vary. The length of the mitochondrion is about 2 microns. The diameter of transport vesicles is 15 nanometers. So these are nanovesicles, nanoparticles, you could say. Vesicles, by the way, are the ones that we find very interesting in terms of transport of viruses. So coronavirus internalization does not take the vesicle transport, but other viruses usually take the vesicle route. Certainly bacteria, intracellular bacteria, take vesicle route. These are endocytic vesicles. In animal cells, this pathway is very important, and we may have some opportunity to discuss it when we talk about motors. What about water? Since everything is made up of water in biology, the volume of water is 10 to the power minus 2 nanometer cube. Density is 1 gram per centimeter cube. Viscosity is 1 centipoise. And the hydrophobic embedding energy, we talked about this in the hydrophobicity part, is 2500 calories per mole per nanometer square. Now with some of these parameters, I don't mind if you don't remember them, but the cellular scales you must, and the organellar scales. DNA, now we come to molecules. DNA has a length per base pair of 0.3 nanometers, 0.34. Volume per base pair is 1 nanometer cube. This is the volume which is made up by the base pair height combined with the diameter of the DNA, which is 2 nanometers. Charge density is 2 electrons per 0.34 nanometers. And persistence length, something we will talk about later, is 50 nanometers. 50 nanometers translates to around 150 base pairs in terms of persistence length. What about proteins? So we've spoken about cells, organelles, water molecules, DNA. What about proteins? So the average radius of a protein is 2 nanometers. Now this is again an average value. As you saw, everything has a distribution. Everything is widely distributed at times in biology, but the average number is 2. So when someone asks you what is the size of a protein, 2 nanometers is a good number. This radius, that means the diameter is 4. Using 4/3 pi r cube, you can calculate the volume of such a protein. It's about 25 nanometer cube. While the mass of an average amino acid is 100 daltons, and the number of amino acids making up a protein result in typical average number, result in 30 kilodalton as the average mass of a whole protein. These numbers, I do expect you to at least have some inkling and not be lost when I ask you these, yeah? Because these are giving us a relative sense of what is the size and shape of the basic molecular building blocks of life, that is DNA, protein, RNA, and lipids. The characteristic force of a protein we'll come about to talk about later is 5 piconewton. The speed of motor proteins is 200 nanometers per second. The diffusion constant of average protein in cytoplasm is 10 microns square per second and can be up to 100. The thickness of a bilayer is 5 nanometers, 2.5 nanometers or 2 nanometers is approximately the height of a single lipid. The area of the lipid is half nanometer square and the mass of the lipid molecule is 800 daltons. So these key facts are nice to keep by you and you may want to refer to them. In terms of estimates, we compare experimental measures to our estimates and we check whether our estimates, we made these estimates of cows and of cells and of number of proteins to accurate experimental measurements. So the molecular census suggests that a cell is a crowded place. But how do you get this molecular census? What method can we use to obtain numbers that we can compare our rough estimates to? And this is the crux of the next section.